Welcome, folks, to Chapter 15, Innate Immunity. Previously, we had talked about various ways for microbial control in the environment, in our food, so on and so forth. But when microbial agents are presented to our body has multiple lines of defense to protect us from those microbial agents. And in fact, usually it does a really good job of protecting us, right? And so we're going to talk about the first couple lines of defense, which are just non-specific barriers that allow for clearance of microbial agents that might be in our environment or might be potential infectious agents. That's chapter 15. In chapter 16, we're going to talk about specifically targeted cells and systems within our immune system that allow us to take out specific pathogens that present to us. So go ahead and pause the video and review previous content, take a look over chapter 15, and then come back. Okay. So this provides us a little bit of an overview of the couple of barriers through which our body kind of protects us from microbial agents.
Host defenses can be divided into two categories: innate or non-specific defenses and adaptive or specific defenses. These defenses act together to protect the host against infection. The human body has many innate or non-specific defenses to prevent disease. These include the first-line defenses such as the skin and mucous membranes, plus the second-line defenses such as phagocytosis, inflammation, and the complement system. All of these defenses act against all pathogens and are not specific to a given pathogen. The third line of defense is comprised of the adaptive or specific defenses. These defenses recognize a specific pathogen and must be activated by antigen-presenting cells before they can work. These defenses include antibodies and cytotoxic cells. Together, these defenses make it very difficult for pathogens to penetrate, colonize, and cause disease.
Okay, so this part will be our focus today. And another way to look at it is to say that this is the science of skin, snot, blood, pus, sweat, and tears, because that's essentially what we're going to cover today. And although many of these things present annoyances to us, and they tend to be problematic to us kind of in our daily lives, the truth is that they are the barriers between us and our environment, and they're activated so that we can kind of keep that barrier and protect our body against incoming potential pathogens. Okay, and so the way that we're going to do this is we're going to kind of alternate between the lecture video, the lecture slides, and outside resources. So I'll just let you know before I start kind of a new section that here are the resource videos. They're all linked below that we're going to be covering before we jump into this section. And you go out to that resource video and then come back into the lecture slides and then go through the slides with me. At the end of the video, at the end of the lecture, once we've gone through the various video tours and the lecture as well, then you're going to jump into the Jamboard and summarize how is it that these guys, skin, snot, blood, pus, sweat, and tears, are protecting us from microbial agents in the environment in a couple of sentences, and you guys can use as many slides as you need for that in the Jamboard. Okay, so let's begin. We'll start with the barrier that protects us from our environment, right? And that's our integumentary system, or our skin. And so just like an individual cell has a cell membrane and a cell wall that keeps it separate from its environment and allows it to organize itself within that space, our skin keeps us separate from our environment and allows us to be organized within that internal space. And so we have different types of organs and tissues and so on and so forth that are all kind of comprised within that. But the skin is that initial layer that provides that barrier for us. That provides that barrier for us between us and the environment, okay? And then there's added layers wherever you have a mucosal membrane, because that's an interface between that barrier and the environment. We're going to talk about those in a minute, but let's start with skin and sweat. And so what I want you to do is pause the video, go to the links below. One says skin, one says sweat. Listen to those. That's about a total of 17 minutes, and then come back into the lecture slides and we'll have a discussion about those. Okay, so as you saw in your video, you have your skin is not just the simple skin that you see at the very top with the epidermis. But what you're looking at is actually a more complex system that has various layers within it. The top uppermost layer is the epidermis. The epidermis consists of both constantly replenished and growing cells that are then pushed up to the very top of the epidermis. And as they get to that top part, they actually die. And they do create kind of like a tightly packed set of layers of dead skin that then also help protect that still living and growing layer that's under it. So the epidermis is kind of a non-living living interface that protects that first layer of skin between you and the environment. Then you have your dermis, which is here. The dermis is alive with glands and various types of capillaries and, your lymphatic system, which we'll talk about in our next video as well. And kind of the interacting with between those cells of the skin and the rest of your body. So bringing in blood flow, oxygen, taking out, so on and so forth, right? Also, it's bringing various types of immune cells to your skin. So one of the things that we have is what's called dendritic cells. And these are types of white blood cells that literally have dendrites. I believe we have or pseudopods, you can see here, dendritic cells, and those pseudopods actually reach in between the various cell layers, and they can actually clean out just generally microbial agents that are in that environment, and they literally eat them. We're going to talk about phagocytosis a little bit later in the lecture today and how that happens, but they do engulf them and eat them. They can also send out signals for the rest of the immune system to pull in other white blood cells as reinforcements. If there's an infection or there's an abrasion of the skin, so on and so forth. We also have the hypodermis, which is essentially kind of an adipose tissue layer that allows for insulation, but it also allows for connection to blood vessels as well. And so you can see here this top layer of the skin. It is that, dead skin cells that are kind of up on the top of the skin and protecting you, again, between the environment. There is this kind of misconception that dust is something like 90% skin cells. And the truth is, if you look at the organic matter components, yes, the organic matter, the components that come off of living things, skin cells are a significant portion of that. But overall, dust particles, skin is a very small portion of dust. Actually, most of dust comes from, depending on the environment, depending on the amount of soot that's in the environment, depending on pollutants, depending on what's going on inside of the house, right? There might be carpet fibers and pet hairs and different types of smoke, if that's part of the environment, different pieces of soil and parts of the environment that are coming in from the outside. But skin cells are actually a small percentage of the overall particles in dust. You see here that this top layer, we said it's a living, non-living interface. So also it needs to be moisturized and it needs to be maintained in terms of its physical structure, but it's also its microbial agents. And so you saw in the discussion of sweat that sweat is produced primarily for evaporative cooling, but it also has antimicrobial agents within it that allow for kind of crowd control of the microbiome. And also there's sebum, which is part of the sweat, which is kind of an oily substance that's primary goal is moisturization of the skin, but this collective of sweat and sebum and its kind of aggregates are also very salty, which we know because we have cultured various types of skin cultures on mannitol salt agar, and that's a salty agar for halophiles. And then there's also different types of dermacidins and lysozymes. Dermacidins are, as the name implies, chemicals that are part of the skin or that are on the skin that are meant to kill pathogens or meant to kill microbial agents. And then also lysozymes are enzymes, actually full-on protein enzymes that will just kind of collect and kill off or eat up microbial agents. Or actually not really eat up, because that's more like a macrophage, what a macrophage is doing, but from a molecular level, actually digest or break down microbial agents. And so these are in and on the skin or on the skin, and they're kind of part of that first line of defense. Now, anytime there is what we call a mucosal membrane, essentially an opening between the skin and the environment. So eyes, nasal passages, mouth, urogenital regions, those have to have extra protection, okay? One, because they don't have that dead skin layer, that epidermis. Instead, they have epithelial membranes or cells. And the difference between those is they're similar types of skin cells, essentially, but the epithelial cell is active in transfer of gas and nutrients and enzyme. It's an active wet membrane versus a dry, just kind of barrier type membrane. And so oftentimes, especially nasal passages, the top of the respiratory tract, there are also cells that actually specifically make mucus in these areas. And so that mucus helps to cover, moisturize, but also to protect those regions against microbial infection. And so that mucus has various types of chemical compounds that help do this. And so what I want you to do is pause the video and watch the outside resource called Mucus, and then come back into the video. It's a couple minutes long. Okay, so you saw that there's different types of antimicrobial agents, as well as even viruses inside of mucus that allow for the inhibition of microbial pathogens, and even collect some of the microbiome within it, right? And kind of maintain the microbiome. There are even organisms that tend to mainly just eat mucus, like Akkermansia, for example, right? That's their main source of nutrients, and they're there and they're part of the natural biome, and they're actually kind of cultivated within mucus. And so here we have these goblet cells that produce the mucus, and we also have ciliated columnar cells, specifically in the respiratory tract and the nasal cavity, that move that mucus out of the body, just like an escalator. So you can see that action here. Okay. It'll take a minute. Here we go. So here we have the goblet cells. You'll see them release mucus in a minute. And then these are those cilia or ciliated cells that are actually moving in a single direction and passing the mucus up the respiratory tract and out. Right? It's very important that the mucus is there to collect, potential pathogens, for example. Any potential pathogens should be moved out of the respiratory tract, and it's those ciliated cells that keep them going in the right direction. Okay? And it is for that reason that when we are sick or there is an infection, that we up-ramp the mucus production so that we essentially can flush those pathogens out of the body and out of our systems. Okay. Similar to mucus are tears, right? So you can go ahead and pause the video and watch the tears video, a simple video that gives you an idea of how kind of tears work and the kind of stages. Okay, so you saw that your lacrimal apparatus, which are your tear ducts, essentially, are producing tears to moisturize your eye and lubricate your eye, but also they have lysozymes and chemicals and antimicrobial agents within them to protect your eyes. Same for chemicals. When you have a chemical, like a chemical or other kind of dust, for example, hazard that comes into the eye, the tears will actually up ramp, and you'll get this kind of second kind of wave of tears to help flush out those chemicals or microbial agents or pieces of dust from the eye and continue to protect the eye. Okay. And then you can see here, then those ducts, the tear ducts, kind of moisten the eye, and then there's ducts under kind of within this nasal region that then collect those tears, and then those tears are gathered back in the throat and essentially swallowed. Okay. We know that the microbiota is an important part of our body, right? So when we talked about mucus, when we talk about skin, when we talk about any kind of part of our body, we have different communities of microbiota. And it's just an important reminder that these organisms are essential parts of our immune system as well, because they metabolize various types of, macromolecules, for example, that are being processed through our body so that they don't build up, so that they don't kind of exist as metabolites for pathogens. They just kind of, take up real estate, right, within and on our body so that pathogens don't really have space to kind of exist. They generate various types of antimicrobial compounds. They also are continuously testing our body's defense system, right? Our immune system. They're continuously testing our body so that the immune system is continuously kind of looking out and vigilant for various types of microbiota. And so it's scanning, it can detect like normal, healthy microbiota, but then it can also still be vigilant if there's a pathogenic or a harmful microbiota. And then also just producing various types of vitamins and so on and so forth that are important for our body as well. So we mentioned this. Okay, so another fluid that's an essential part of our body is blood. We will actually do a blood video that's also a summary of the lecture at the end of the lecture, so stay tuned for that. Blood is a matrix, both red blood cells, white blood cells, platelets, various chemicals, for example, those chemicals that are part of the complement system. And so it also has salts and electrolytes. Electrolytes are essentially just salts or ions that are kind of dissolved within the blood. And then it also has the living components. So you have your red blood cells and your white blood cells. Those are living cells. And then you also have platelets, which are pieces of dead cells. So they were previously living, but they're not living now. Platelets are essential to clotting, right? And so it's important that if there's abrasion in the skin, blood is not lost. And so it's important that it has the components that it needs to actually clot or kind of maintain that barrier if the skin is perturbed. And so platelets are important to that. Another thing that blood does for us is the movement of nutrients throughout the body. And so you'll see, if you remember back to sweat is essentially coming off of blood. It's essentially parts of the plasma of the blood, as is when we talk in chapter 16 about lymph in the lymphatic system, which is a liquid that is also coming off of blood. And so it has in it many different components of our life and our health within that plasma and within that blood. Then kind of our focus today is leukocytes. So you can see here we have our red blood cells and our platelets, but then all of these, 1, 2, 3, 4, 5, are different types of white blood cells. So this first type, basophil cells, are the only ones that do not become phagocytic. And essentially, we're going to talk about phagocytosis in a little bit, but those are the cells that we had mentioned before that can actually engulf and actually digest microbial agents. Instead, basophil cells, they send out the chemical signals that say that there is a breach of the skin, for example, or a potential pathogen, and they actually, those chemicals actually bring about inflammation. We're going to talk about inflammation and the importance of inflammation to recruit white blood cells and also kind of to isolate the area of whatever the pathogen is or the abrasion to the skin, okay? Then all of these neutrophils, eosinophils, monocytes, lymphocytes, all of these do phagocytose, okay? They also send out chemicals to recruit and reinforce when necessary, but they essentially will, take on and eat or engulf microbial agents that are coming into the body and digest them. Now, lymphocytes have 3 different types that they can mature into. One is a natural killer cell, and these are general white cells that kind of just take out microbial agents generally. But also lymphocytes, the other 2 types, are T cells and B cells, and these are very specific immune cells that detect very specific antigens. And so T cells and B cells are going to be a big portion of our discussion in chapter 16 as well. Okay. Okay. And so these different immune cells, these white blood cells that are helping us take out microbial pathogens or microbial agents that might be dangerous to the body, they show up whenever there's a potential infection or abrasion to the skin or other areas of the body. And so we can actually measure some of these cells, and it gives us an idea of maybe what the potential pathogen actually is. So for example, eosinophils tend to be a reaction to parasitic worms more than a reaction to bacterial or viral infections, whereas just generally white blood cells, but specifically neutrophils, are more of a reaction to bacterial pathogens. And then lymphocytes tend to be more kind of the reaction of the body to viral infections. So it's not foolproof, but it does give you an idea of what the potential pathogen is. It is also the reason, the recruitment of white blood cells, that is, the reason why we get pus when there are infections within the body. So there's actually just a very short, simple video. There are a lot of videos that were just a little bit too, I don't know, overwhelming or inappropriate, I guess. So just a really short video on pus. So go ahead and pause the video, watch that one, and come back in. Okay. So you can see that when there's an infection, these white blood cells are coming in drones and trying to, address that infection before it spreads throughout the body. And this is sometimes why we get pus. Okay. Once white blood cells get to this area, specifically those that are not basophil cells, and they can actually phagocytose, what are they doing? Well, it's pretty interesting. Just the agility and the mobility of these cells, that they actually have these pseudopods that can reach out and grab onto pathogens and microbial agents and ingest them. And so one very similar or one organism that has a very similar mechanism for eating is a unicellular predator called amoeba. And so you may have heard of amoeba before, but you're actually going to be able to see the various stages of phagocytosis. And so those stages are chemotaxis, where you're actually the predator. In the case that I'm going to show you with the amoeba in our body, it's these white blood cells are actually moving towards whatever that agent is, right? That it's going to engulf. Adhesion, where it actually connects via receptors to whatever the molecule is, either the microbial pathogen, or in the case of the amoeba, it's lunch. And then it engulfs via that adhesion, it actually engulfs the particle, and it creates a vessel around, or a vesicle around that particle. And then it fuses to those vesicles lysozymes, which are digestive vesicles. And then those enzymes that are released from the lysozymes essentially digest that pathogen or that lunch particle. And then sometimes the particles are eliminated by white blood cells, and sometimes they're actually retained. It just depends on the type of white blood cell. So you can see that here. This is the amoeba, not the white blood cell, but it's a good example. Okay, so chemotaxis, where it's moving toward, this is a little paramecium, and that's going to be engulfed. And those pseudopods, this is real time. Pseudopods move very quickly. And then you'll start to see that the vesicle is formed 3D. So the membrane is going to start to close off at the top and the bottom of this field of view. And that's going to create a vesicle around that protists. Okay, so you can see the membrane forming here. And then on the bottom of the slide there, it's going to cover and actually completely engulf this protist. And then what you can't see exactly in this image is that some of these vesicles that are surrounding the vesicle that's being formed around this protist are then releasing enzymes. They're lysosomes that are actually releasing enzymes and acids into that vesicle, and then that protist will actually be digested. In the case of the amoeba, it actually ingests and retains those nutrients. In the case of white blood cells, it depends on which white blood cell that you're talking about. If you're talking about neutrophils tend to kind of ingest and actually just, at one point they just die. Whereas other types of white blood cells will actually sometimes release those particles of the pathogen that they've ingested. It just depends on the scenario and the type of cell. Here you can see a video of phagocytosis that's a little bit more specific to white blood cells as well.
The GMC AT4 lineup, premium and capable.
Sorry about that. The other video was still playing.
Of a cell toward or away. Chemotaxis is the movement of a cell toward or away from a chemical stimulus. Phagocytes use pseudopods to move toward microorganisms or damaged cells at the site of infection, often arriving only minutes after infection. Chemotactic chemicals that attract phagocytes include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides derived from the complement system. The plasma membrane of phagocytes usually attaches to glycoproteins on the microorganism's surface. This process is called adherence. Adherence is made easier through opsonization, a process in which antibodies or complement proteins from the host coat the microbe's surface, serving as handles so that the phagocyte can more easily attach to the microorganism. The proteins that coat the microbe are called opsonins. The pseudopods fuse, forming a sac called a phagosome. The phagosome enters the cytoplasm, where it fuses with lysosomes that contain digestive enzymes and other antimicrobial substances. The fused structure is called a phagolysosome. Digestion of most bacteria is complete within 10 to 30 minutes. After phagocytosis, the remaining debris is eliminated from the cell by exocytosis, in which the phagolysosome fuses with the plasma membrane and expels its contents. Some of the contents of the phagolysosome may also be presented on the cell membrane through a process called antigen processing and presentation. Let's review the steps in phagocytosis. The pseudopods of a phagocyte use chemotaxis to approach the microorganism. The pseudopods adhere to opsonins that are attached to the phagocyte. The microbe is ingested within a phagosome. The phagosome fuses with a lysosome to form a phagolysosome, in which digestive enzymes break apart the microorganism. Finally, the remaining debris is eliminated by exocytosis. Parts of the microorganism are presented on the surface of the phagosome.
So we'll talk more about antigen presentation when we talk about the adaptive immune system in chapter 16, but it's really interesting that essentially these cells kind of wear the parts of those microbial agents that they decompose on the outside of their cells. Not all of them, but some white blood cells do, so that the body then knows that these are potential pathogens later on. Okay. So in terms of the body's response to pathogens as they come in, there are a number of different chemicals that are part of the blood and released via various white blood cells. We're just going to focus on two, a process called interferons, which kind of promote a process for cells to essentially recognize that they have a viral infection, and then the complement system, which allows for inflammation of a specific area, which is essentially like a quarantine and also recruitment of white blood cells. Okay. And so here, in terms of interferons, essentially these are chemicals that are released by white blood cells that are infected by viruses, kind of like a warning signal that's released and then spread throughout the blood and into the surrounding tissue to surrounding cells as well. And essentially, one of the unique issues with viruses is that since they actually hijack the host's machinery, if you are to close down or shut down that machinery, then whatever chemical it is will actually affect other cells within your body as well. So it can be difficult to get at viral infections for that reason, because they essentially are presenting in a very similar way to how your cells normally function. And so interferons, essentially, once a white blood cell is infected, it releases these chemicals warning other surrounding cells that an infection might be coming, right? If that virus replicates within that cell and then through the lytic cycle produces many of itself and is released, those are released into the environment, then they're coming, essentially. It says that there's an infection, potential infection that's coming. And what that does is the interferons then make a chemical cascade that actually prevents the cells from doing some of their normal functions related to transcription and translation, because that's what the virus is going to hijack, right? It's going to hijack transcription and translation, and it's going to try to utilize those mechanisms. So it does slow down the cell's own processing. Let me give you an image that you can see here, right? So it does impact the cell's own processing because it doesn't do transcription and translation, kind of a general kind of overall halt to transcription and translation for a couple days or a couple hours, however long it takes to kind of clear that infection. It's a couple hours, but it might continue through various cells for a couple days. So just to clarify that. And so one of the chemicals that is an important part of this mechanism is these antiviral or AVP proteins that just exist within the cells all the time. But when an interferon actually activates them, then sometimes they will start destroying RNA, just like destroying RNA molecules helter-skelter. Some of those are your own RNA molecules, but if a virus was to infect, then the idea is that it would also get the viral RNA before those actually were converted into proteins and then activated. So it allows for the cells to kind of essentially warn surrounding cells and prevent infection in surrounding cells. So that's one example. The other example of chemicals that are in the blood or that kind of allow for us to respond to pathogens is a really important system called the complement system. And the complement system is a series of various proteins, very simply named C1 through C9, right? That always exist in our blood, but they can be activated through various mechanisms, usually related to white blood cells. And what they do is they start to just generate a response specifically around inflammation. So inflammation can sound like a really negative thing sometimes when you have, inflammation on your skin, But that inflammation, what it's doing is essentially quarantining a threat. Maybe it's a cut, or maybe it's a pathogen or an infection, quarantining that threat and calling in, recruiting other white blood cells to that area. And so these two videos, there's some complexity and there's different pathways to the complement system. You don't need to know the specific proteins or the specific pathways, but understand what the complement system does and what it kind of elicits in the body. Okay, so you can see that.
The complement system is a set of proteins that circulate in the blood serum. When activated, these proteins work together to destroy foreign cells by cytolysis, to activate the inflammatory response, and to assist in phagocytosis. The main components of the complement system are called C1 through C9. Once the complement cascade is activated, several events occur that ultimately result in the destruction of the pathogen. C3a and C5a act as chemotactic factors, attracting phagocytes to the site of infection. C3a and C5a also bind to mast cells, triggering the release of histamine and other chemicals as part of inflammation. C3b acts as an opsonin, coating the invading cell. This coat makes it easier for the phagocyte to adhere to the invading cell during phagocytosis. C5b binds to C6 and C7, which attach to the plasma membrane of the invading cell. C8 and several C9 molecules bind to the C5b, C6, C7 complex, forming a cylinder-shaped membrane attack complex, or MAC. The MACs form circular holes in the membrane of the invading pathogen, causing water to enter the cell and ions to leave. This results in cytolysis, killing the cell.
Okay. Okay. So these various kind of cascades, it's also called the complement cascade, allows for inflammation. Well, let's talk a little bit more about what inflammation is and why it's important, okay? And so you can see here some information about inflammation.
Damage to the body's tissues through heat, chemicals, sunburn, cuts, or microbial infections triggers a general, nonspecific defensive response called inflammation. Signs and symptoms of inflammation include redness, pain, heat, swelling, and sometimes loss of function. Inflammation has three main functions: to destroy the agent causing injury, to limit the effects of the agent on the rest of the body, and to repair or replace damaged tissue.
Okay, and so this vasodilation that's occurring right here is really essential because that's how our white blood cells are actually even getting to the location of the inflammation or the infection. So there's a little bit more information here.
Damaged cells release several chemicals, such as histamines, prostaglandins, and leukotrienes. Certain components of the complement system can also stimulate the release of histamines. One function of these chemicals is to cause vasodilation, an increase in the diameter of arteries and arterioles. Vasodilation results in more blood, and therefore more phagocytes, oxygen, and nutrients being delivered to the site of injury. This increased blood flow results in redness, heat, and some of the pain associated with inflammation. The histamines, prostaglandins, and leukotrienes also have a direct effect on the capillaries in the area of the injury. They cause the walls of the capillaries to open and become leakier. This allows more fluid to leave the capillary and enter the tissue spaces, causing swelling or edema. The increased blood flow delivers phagocytes, such as monocytes and neutrophils, to the scene. The phagocytes stick to the walls of the blood vessels in a process called margination, then squeeze through the gaps between the cells in a process called emigration or diathedesis. Within about an hour of tissue damage, the phagocytes begin to destroy the invading microorganisms by phagocytosis. After engulfing large numbers of microorganisms and damaged tissue, the phagocytes die, forming pus. Other components of the blood lead to the formation of blood clots, which seal off the site of injury. The final stage of inflammation is tissue repair. The increased delivery of nutrients and oxygen from vasodilation aids in this process.
So you could have acute inflammation, which just allows your body to kind of take care of that site of infection or that abrasion and kind of deal with it and heal and so on and so forth. But in the case of chronic inflammation or inflammation due to secondary infections, that's when sometimes you can actually have issues and kind of long-term tissue damage and so on and so forth. So it is important that inflammation occurs, but it's also important that it kind of passes and then that tissue is able to be repaired. Another example of these symptoms and signs of infection or disease is fever. So what's going on with fever? Essentially with fever, you have chemicals within your body that tell your body that there is a pathogen that's infected or kind of breached the body's barriers. And when these chemicals are present, then the body responds with its various lines of, immunity and the various white blood cells, so on and so forth. But sometimes it's just not enough. And those pathogens continue to replicate, and those chemicals that indicate the presence of those pathogens are continuing to be present. In that case, your body actually sends a message to your brain that says, okay, light it up, right? And that increased temperature, there's a couple of different goals with that. One is to allow for your white blood cells to kind of carry out their actions as efficiently as possible. So that includes these histamines, for example, and cytokines that are chemicals that are released so that the white blood cells can communicate with one another. They're going to be more quickly spread through the body if the temperature is increased. Also, just the fact that microorganisms, pathogens of our body, are going to be at homeostasis at our body temperature. So if we increase the body temperature, just that increase in body temperature allows us to kind of put them at a state that's not in homeostasis, and then actually may allow us to clear them in that way. Okay. And so in those ways, fever, and we don't actually understand all of the mechanisms of how fever happens and how, it's communicated to our brain and so on and so forth. But we do know that it has multiple kind of ways in which it can kind of help us clear those infections within our body. Okay. And so you can see that here. And so same as inflammation. If fever is short-term, it's able to allow for those chemicals to circulate between our different parts of our immune system and through our lymphatic system and kind of, communicate, a response to that particular infection, then it's great. But when fever is prolonged, then you start to see your own body, which is at homeostasis at body temperature, start to deteriorate. And so secondary infections or prolonged fevers can become a really big issue. And so it is important that our body is able to respond with fever, but then efficiently clear the infection and then move away from that fever as well. So essentially, move this over here for a second. Essentially, those are the mechanisms by which our body is responding to kind of pathogens that present themselves in our environment and to our body, right? And so those responses allow us that first line of defense to try to clear our body of pathogens. But then there are particular specific kind of lines of immunity that we also need to have for specific pathogens that present a higher risk to our body. And so in chapter 16, we're going to talk about adaptive immunity. specifically focusing around those T and B cells of the immune system that we had kind of just glossed over and mentioned in today's lecture. So the final video in our video tour is a crash course that kind of overviews innate immunity. And so go ahead and watch that crash course video and then go through reviewing your notes and then jump into the Jamboard and give a summary of how skin, snot, blood, sweat, and tears are our first line of defense against pathogens in our environment. Thank you for listening, and I'll see you in the next video.